Okay, I think it's working. All right, this is going to be chapter 13. I thought this was up, but it's not. Um, I tried to put the PowerPoint up, but they're telling me it exceeds my quota. So what I have to do at this point, I think, is take a deep breath, run through the PowerPoint, and then I'll embed it into um, YouTube, and then I'll embed it into the course under 13. So this will be a kind of an introduction to the immune system. I want to spend some time on the immune system. Like I said before, I think if you had me for lab or whatever, a lot of instructors tend to ignore this, but I think it's extremely important, especially if you're going to healthcare. And I'm assuming you're going to healthcare if you're taking this course. So I want to go over just some basics. Most of this is from McGraw-Hill. Some of them might be from Pearson. I have embedded some videos, so I'm hoping they play. If not, I can try to put them in separately. I'll do my best, but ever since the last year or two, we've had like no storage, which is just aggravating, but we'll get through it. So I'll just start here. The human body exists in a state of dynamic equilibrium. Okay, so that literally means that nothing is static. Your body is in complete equilibrium. We were talking in lab yesterday. We did our throat swabs and our skin swabs, and some of you were a little freaked out about the staph aureus or staph epi growing on the body, but I said you really want this there. Okay. Staph can grow because it can grow in a higher halophile. It can grow in a higher salt concentration, and your skin has, an acid mantle and a little bit of a salt mantle, which inhibits some bacteria. If you don't believe me, get someone's permission. You could lick them. They might be a little salty, but you'll get the gist of the salt content of the skin. And then, like I said, that inhibits most other bacteria, but it will allow staph, epi and staph aureus to grow. And then we had a really good point. They said, well, what about a staph infection? Okay. So the staph has to breach your first line of defense. So that's going to be your skin, your mucous membranes, your tears, saliva. It has to breach that before it can get into your basal membrane, into your capillary bed, or into your bloodstream, or you have to have an open cut. And then we just tested some disinfectants, and I think we had the discussion about hand sanitizers. I see some people have to use them, but there's a lot of alcohol in a lot of those. It tends to dry your skin out and make it crack. All right, and so then some of these bacteria have a nice little crevice to get in and grow. So if you need to do it, you need to do it. Washing your hands with soap and water, drying them is a good way of doing it. We used to do a lab where you would just touch your slide. We take a petri dish and we divide it in 5 or 6 quadrants. You touch it with your finger, then you would rinse it with your finger, touch another section, then you would wash your finger, rinse it, touch it, then you would dry your finger and touch it again, and then we'd see each one of those methods had less and less microbes on it. But I'm digressing. All right. And so many interactions between human body and microorganisms involve the development of biofilms. And I think we touched upon that earlier. And I know some of you in lab are in dental hygiene, so we were talking a little bit about that. But just realize this is where bacteria finds a niche. It lays down a foundation, and then through quorum sensing, it'll decide whether it's a good environment, and it'll start replicating. And then as it replicates and ferments whatever, or changes the environment, it changes the oxygen concentration higher or lower, depending on what's going on. And other organisms come along, they find a niche, and they grow on top of that. And after a while, you can have 5 or 6 different organisms growing. All right. And then the cool thing about that, if you're a bacteria, is if you're in one of the inner layers, you're protected from phagocytosis from the immune system and some of the antimicrobials. And I know I'm almost positive in there. There should be a video on biofilm. All right, it's a 3D animation of how it grows. So just if you can find it, refresh yourself or just pop on YouTube and see should be an animation biofilm. Just see how they grow, and they're finding out, this is growing on PICC lines and catheters, and can be a real problematic in healthcare. All right, so colonization of the body involves a constant give and take, and we could have the argument that certain parts of the body are sterile or close to sterile, and then even with birth, the baby's going to get their first inoculation if they're vaginal birth through the birth canal. Right. So let's see if I can get this to work now. All right. So microbes that engage in mutual or commensal association, these are your normal resident flora, indigenous flora microbiota, right? So please realize we do want this, and I talked about it in lab. We want some inhibitory bacteria on the skin so that nothing more pathological, more virulent can grow, all right? And we're not talking about digestion here, but please realize, I know I have a discussion in the course about the microbiome, and they're really finding out. I know, guys, it's a jam-packed summer, but when you're done, please start researching the microbiome and how a lot of these microbes in your gut are communicating with your brain through the gut-brain axis, through the vagus nerve, and they're finding out through research that a lot of these microbiomes are really deciding your emotions, what you crave, and things like that. And there's proof where people have had their microbiome wiped out, and they had a fecal transplant, or they had somebody else's microbiome introduced, and they literally changed all their food cravings, their emotions. Not to digress, but there's stories about people who've had, similarly related. They've had heart transplants or organ transplants, and they've taken on the traits or the cravings or desires of people that they had the organ transplant from. All right, so there's a little bit more to it than what we should think with biology dogma. Sorry, his allergies are horrible. It's mostly in this room. I don't know what is going on, but infection is a condition in which pathogenic microbes penetrate host defenses, enter tissues, and multiply. And that was a question I got in lab. Well, what about staph being on your skin as normal flora? That's great, but what we don't want is we don't want that multiplying inside the body. Because I talked to you guys yesterday about staph, and we haven't gotten to it in the lecture, but I said it has many virulence factors. So it can produce enzymes to deal with oxygen, red blood cells, white blood cells, coagulase, hyaluronidase. It can eat through, pretty much any portion of the body. And I said, kind of joking, I said, if you guys want to come back as a bacteria, you want to come back as Staph aureus. And I said, you want to come back as MRSA or vancomycin resistant, something that's really resistant to everything we're throwing at it. Right. So pathogen is an infectious agent. And when we say a pathogen, we're saying that something that's going to cause a pathogenic disease or problem, because some things are innocuous, more or less, unless you're immune compromised or something else is going on. Then that can turn into a pathogen, something that would be innocuous or normal flora, like candida or something. That's normal, but if you're immune compromised, it can cause a problem. And we have a problem right now with Clostridium difficile, which I talked to you guys in lab is a normal part of your flora for 40% of the population of the large intestine because of the decreased oxygen concentration. But if you wipe out the microbiome and it has no competition, that's when it causes all the major problems. And if you're going into healthcare or you're any nursing home, or if you're LPN or whatever you're doing right now, you definitely have heard of this before. So now an infectious disease is an infection that causes damage or disruption to tissues or organs. So you can have something more or less growing in your body. As long as it's not destroying or disrupting anything, it's not going to be a problem. But when it starts taking over and starts destroying your mucous membranes or subcutaneous tissue or things like that, then we have an infectious disease. All right, so here's contact, colonization, infection, disease, and it's just really showing you. I think this is from Pearson. I put it in here because it's kind of a really no, this is McGraw-Hill. Sorry. We have to have contact, all right? But the microbes adhere to the exposed body surfaces. Now that can be through fimbriae, it can be through slime layers, it can be just stuck in grooves of your skin from hand sanitizers and things like that. So we have to have contact, then we have to have colonization. And I said in lab the other day, I said, everyone thinks that they're, center of the universe, is kind of joking, but please realize a lot of these microbes have nothing to do with you. You're not going to be a hospitable host for them, or you're not going to be a good fit for them. So you can have contact with them, but if they're not going to colonize, or if the pH isn't right, or the oxygen concentration is not right, they're not going to bother you, right? Right, just like a lot of these bacteria that are have niche markets, or they're haliophiles or thermophiles or cryophiles, depending on the temperature, they're not going to find you hospitable. And I was talking to somebody this morning, actually, at LA Fitness, and they said, is the steam room going to be open, or the sauna going to be open? I said, well, I don't know. It depends. They could say COVID, they're not going to open it. But, I've had the discussion with people. I said, listen, there's no human pathogen bacteria that's going to live in that environment. All right. So the bacteria that are going to live in that environment are not going to find you hospitable. Your body temperature is too low. Anything that's going to live in there. So, you can have that discussion. So invasion is microbes cross lines of defense and enter sterile tissue. So you have to have contact. It has to colonize, reproduce through binary fission, and then it has to be able to invade your tissues to get inside. If it's growing on your skin, not going to be a major problem. There are some molds and mycosis that can grow on there, but for the most part, not going to be a major problem. Infection is when the pathogenic microbes multiply in the tissues. So you could have a skin infection on the exterior, but usually when we talk about infection, it's when something that's penetrated and gets in the tissues, and then God forbid gets past the basement membrane and starts multiplying in the bloodstream. So carrier state develops. Microbes are established in tissue, but disease is not apparent. So these can be multiplying, but until they start releasing enzymes, exoenzymes or endogenous enzymes, or we'll see gram positive, gram negative release different exotoxins or endotoxins. Unless they're releasing something, or they're in the tissues and then they're releasing something, they're probably not going to cause a disease. They might just be inconvenient. Defenses hold pathogen in check. Usually, effects of the microbes result in injury or disruption to tissue. So it depends on what they're releasing in their environment. So whether they are fermenting the sugar on your teeth, or if they are staph and they're releasing digestive enzymes to eat their way through whatever. We can have morbidity or mortality occur, and just different effects depending on what's going on with that specific pathogen or potential pathogen, and how your immune system is and how you're handling it. But we really want to make sure that if we're inhaling it, it's in the nasal mucosa. If it's mucus, we have IgA. And as long as we're not getting something up into the brain or past the meninges, we usually can deal with it. And I was talking to somebody yesterday in lab about, what is the difference between the upper respiratory tract and lower respiratory tract, and why does strep want to grow on the back of your throat? Why wouldn't it grow other places? And, we really want to get into body temperature, moisture, oxygen concentration. Things have to be just right. There's got to be, a source of food for them. And we talked a little bit about alpha and beta hemolytic strep, and how it's going to change that blood agar plate and change it to clear or yellow or greenish, depending on whether there was hemolysis, or lysis of the red blood cells. And we talked about, how your body likes to sequester iron, because bacteria need iron to multiply. So your body can sequester that, usually in the liver, but some forms of bacteria can go and they just start lysing red blood cells, and they just steal the iron, off the hemoglobin. right? So I said before, bacteria are way smarter than we are. They're going to find a way. So resident flora are most areas of the body in contact with the outside environment harbor resident microbes. And yesterday we did our mannitol salt swabs. We said was differential and selective for staph. We talked about the color change for Staph aureus, for Staph epi, and the colonies. Staph aureus means golden or yellow, so those colonies should be yellow. They should also ferment the mannitol alcohol sugar. And Staph epi is going to be a white colony. All right, and those should not change the agar color. And then we did our throat swabs. So please realize we have alpha beta hemolytic strep. You could have Neisseria, diphtherides, you could have candida. So especially if we're all working in healthcare, you could have things growing in the back of your throat. Don't get freaked out. It's normal flora. And like I said, you kind of want that so that anything else that you inhale or come in contact with, it's got some competition so it's not just spreading wildly. So internal organs, tissues, and fluids are microbe free, or they should be. We can get into the discussion on whether your stomach is sterile or your lungs are sterile or certain areas of your body are sterile or supposed to be sterile. There's all kinds of factors that play in that. So transients are microbes that occupy the body for only short periods of time. And we're talking usually transient microbes are what's growing on your body. So as long as you're bathing or showering occasionally, that's fine. And we talked about Aticept. I think I have right here. So it has triclocan in it. So people that have major issues with that, there are soaps that have antimicrobial properties, or you could take a salt bath that will kill most things, even staph or strep if the salt concentration is high enough. And residents are microbes that become established. So you can have transient, but if you have something that's transient and you're not cleaning it off, or it overcomes tissue, or if you're, I don't know, a heavy sweater and you've got a lot of food, or like staph likes pores and hair follicles and things, they can become very established. So sites that harbor normal resident microbes. So this is normal, all right? Skin and its contiguous mucous membrane. So we had you swipe the nares of your nose. So that is your skin and the connecting contiguous mucous membranes. Upper respiratory tract, talked about that yesterday in lab. Gastrointestinal tract, various portions of it. And just think, things can get past the stomach acid. All right, and they can get in the duodenum, and depending on what portion of the GI tract they're in, we'll talk a little bit later about cholera, salmonella, some of these other GI tract issues. And at the end of the semester, we're going to be doing the Enterobacter ACA lab, and we'll deal with E. coli, Pseudomonas, Klebsiella, Proteus, some of these things that are or could be normal GI microbes. A couple semesters back, we had somebody who had reptiles and had salmonella, and that can be part of your GI tract. It's on chicken eggs, and it can be on turtles and reptiles and things, and you can inadvertently digest that. So we'll briefly talk about that. Outer opening of the urethra, right? And we'll talk later about urinary tract infections, and in males and females, it can go either way. It can happen external genitalia, vagina, external ear and canal, external eye, lids, and lash follicles. And we talked about the dermal decks, those lash mites. So remember, once again, if you're using a microscope, please wipe the oculars off with alcohol. External ear and canal. And a lot of you were swabbing your inner ear. you might have something growing in there, but the wax and cerumen in the ear usually inhibits most. So you kind of want to do maybe the outer lobe or behind the ear if you want to see what resident flora is there. So resident flora is bacteria. Flora benefit hosts by preventing overgrowth of harmful microbes. All right, so once again, we want those there. We want to have inhibition for other microbes. Endogenous or endo means inner infections occur when normal flora is introduced to a site that was previously sterile. So as we get in to the end of the course, we'll be talking about the pathogenic, gram-negative, gram-positive, cocci, diplococci, Neisseria, gram-negative, like the double D shaped organism. And we're going to realize that E. coli or some of these Enterobacteriaceae bacteria are fine in your GI tract. It's when you introduce them to the urinary tract or the reproductive tract or the eyes or the lungs where they can start becoming problematic. Or initial colonization of a newborn, and it just shows you here, vaginal birth. All right, and then subsequently people are touching the baby and inoculating the baby. All right, so if you have a newborn, I personally right now I wouldn't let a lot of people be touching my child unless they were washing their hands. We want to inoculate the the infant slowly with parents' normal flora. All right, uterus and contents are normally sterile, remain so until just before birth, and we'll talk about saprophytes, some of these bacteria that are normal for mom, and they usually don't cause a problem. It's when mom has some kind of STD or chlamydia, we want to really be flushing the eyes out or making sure we're getting rid of that doesn't cause any blindness. So breaking a fetal membrane exposes the infant. All subsequent handling feeding continue to introduce what will be normal flora. And we could have the discussion on breastfeeding and how mom can give immunoglobulins to the baby, and we want to introduce that baby to the microbiome of the parents or the surroundings. Right. So there's indigenous flora of specific regions. So just realize you can look through this and realize like candida can cause thrush, but that's immune compromised. So you're going to have that in your mouth. Women have it in the lower reproductive tract, right? Oral cavity. We looked for streptococcus yesterday. Like I said, in the lab, you could have Neisseria, and it was like, oh, Neisseria gonorrhea. Well, yeah, it doesn't necessarily mean it's the same thing, or if it's in the back of your throat, it's one thing. If it's growing somewhere else south, it could be a different thing altogether. Lactobacillus is normal. Bacteroides are normal. So any of these, Streptomonas, right, species of syphilis, Haemophilus, what they used to think was cause of flu, is there. These are all normal. So on your lab, if they ask you name 4 bacteria that could be normal flora of the throat. So these are all normal. Your skin, staph, micrococcus. So that's going to look gram-positive cocci. It's going to look very similar to staph or strep. Corneobacterium, propionibacterium causes acne, streptococcus. So we'll be learning about these as we go along, but just realize some of these things are normal for those areas. It's when they get into other areas that they become a real problem. Right. So flora of the human skin. Skin is the largest and most accessible organ. It is your largest organ. Right. 2 cutaneous populations. So transients, once again, are influenced by hygiene. So these are on your skin as you shower, or please realize you're shedding skin. Your skin is literally dead. What you're looking at is dead. We want that there for a reason, because we're shedding it constantly along with the bacteria. Right? And residents who are stable, predictable, less influenced by hygiene. So those would be things that we are on our skin continuously or more continuously. All right. So GI tract is a long, hollow tube bound by mucous membranes. The tube is exposed to the environment. And we could have the argument your GI tract, respiratory tract are literally outside your body. They're all facing the outside. Anything that has a mucous membrane is literally outside of your body. So you need to just wrap your head around that. So variations in flora should be due to shifting concentrations. And I talked about this in lab. Your pH is different. You know, from your stomach, pH could be, let's just say it's a 4. And as you go along, it changes. And then oxygen concentration, oxygen tension. As you go further, all right, further through, the oxygen concentration gets much more diminished. So a lot of the anaerobic Clostridium difficile and a lot of the anaerobic microbes want to be further down the tract because there's less oxygen. Oral cavity, large intestine, and rectum harbor appreciable flora. Oral cavity is, you know, telling you guys in lab, I think, third lab or whatever, I'm talking about microbiome, and I told you guys, dogs' mouths are usually cleaner than humans. Right? You have over 500 different types of microbes in your mouth. Large intestine is huge, appreciable flora. Flora of the mouth is most diverse and unique flora of the body, numerous adaptive niches. All right, so like I said, these are going to have something going to attach to your teeth. They're going to start fermenting the carbohydrates or sugars in your teeth, and they start producing acid. All right, changes the pH, and then something else comes along and likes that pH change and the oxygen change, and then you have all of these biofilms growing. And if you don't believe me, brush your teeth at 7, and by 11 o'clock, teeth are gonna feel different, right? So there's biofilm growing. Just think of the bacteria count in just your saliva. A human bite can be septic, and we talked earlier about rinsing or gargling with hydrogen peroxide can really diminish a lot of these organisms. Right. Flow of the large intestine has complex, profound interactions with the host. Once again, you need all these, especially vitamin K production, things like that. Intestinal environment favors anaerobic bacteria because of the lower concentration of oxygen, more distal. Intestinal bacteria contribute to intestinal odor, skeatal, it's called S-K-O-T, something like that, I can't remember. Right. For the respiratory tract, oral streptococci, which is what we were looking for sort of yesterday when we did our throat swabs, we'll know tonight. First organisms to colonize, nasal entrance, the vestibule, all right, Staph aureus. So hopefully we're going to get some of those on our mannitol plates. Mucous membranes, the nasal pharynx, Neisseria. I remember when I first started teaching micro here, I did my throat swab, and Dr. Rowe was like, "Oh yeah, that looks like Neisseria." I'm like, "What?" She's like, "Don't worry, it's normal." All right, but you know, I see a lot of patients, and you know, exposed to different microbes. Tonsils and lower pharynx, Haemophilus, influenza. All right. So maybe. All right, so you guys can watch this on your own. It literally, Mythbusters literally talks about, you know, a hanky elbow or Kleenex, which is the best way for a sneeze. Just talk about nasal hygiene. So watch that if you get a chance. Pass this somehow. Please don't tell me I'm going to watch this for the 20th time. Because the guy has green stuff on his mustache and it makes me just want to gag. I don't know how to fix this. Next. All right, sorry guys. Let me just.
Testing some techniques for stopping sneezes. Let's get this skin. Now you know. Help me go. You look like a pretty strange.
Oh my god. Oh, I just had it. All right. So flora of the genital urinary tract. And just realize this is where we can get some of the urinary tract infections. And if we have something that has pili, it can attach, or pili and fimbriae, it can start traveling up the urethra, up the ureters, and then if it gets into the kidney, we can have some hydronephrosis, and some of these bad kidney issues. So sites that harbor microflora are females, the vagina and the outer opening of the urethra, males, anterior urethra. Changes in physiology influence the composition of the normal flora. So the vagina, estrogen, glycogen, and pH, so that changes hormonally. Women can have change the pH or the sugar content, glycogen. We can start having candida, yeast infections, things that normally wouldn't be there. But any alteration in the environment can change things. It makes another niche market for something else. So normal flora is essential to the health of humans. Flora create an environment that may prevent infections and can enhance host defenses. So we want that, because remember, your organism is they're gonna be recognizing pathogen-associated molecular patterns. So even if it doesn't specifically recognize whatever that is, there are certain molecular patterns that certain organisms have. So if we have something that's similar to it, and we have a microbe that comes along and has some kind of similar pattern to it, the immune system will already attack it. It may not be specific for that organism, but it realizes that that is similar to something else, and it knows it's not self. Antibiotics, dietary changes, and disease may alter the flora. Antibiotics are huge, so please start investigating antimicrobials and how they're wiping out the normal flora, and that's why we have the big C. diff issue right now, because remember, Clostridium is a spore former, so we can kill the vegetative cells, and just the spores are going to be there, and we already know that those can come back. Right. Probiotics introduce known microbes back into the body. So kombucha, you do fecal transplants, yogurt sort of kind of works. Are major factors in the development of an infection. So we always have to have a portal of entry. So whether that is respiratory, mucus to the mouth, can be the urinary tract, can be the reproductive tract, could be the other end too. Anything's possible. The microbe has to attach to the host cells. Microbes must make a pathway into the cells. They have to find you hospitable. They have to find a way. They have to make everything as hospitable. They have to realize that the environment's good, or they just start reproducing. All right. Microbes attack specific tissues, and it's going to attack specific tissues that are hospitable to them. So just realize that not all bacteria are going to live in all different places, because the pH or oxygen concentration or sugar content or whatever, just like viruses cannot just attack any old cell. They have to be very, very specific. All right. So old age and extreme youth, as far as the immune system, is not up and running in an infant. We could have a discussion about immunizing children under a few months old. I don't know. Probably get in trouble for saying it, but I don't know why we're doing that if these infants can't actually produce antibodies. So it doesn't make any sense to do that. Infant can't produce antibodies. So what is your rationale behind vaccinating someone that young? Genetic defects in immunity and acquired defects in immunity. So we can talk about the George's syndrome, or having a problem with B cell production, T cell production, thymus issues. Surgery or organ transplants can diminish that. Organ transplants, we have to definitely diminish the immune system, so we don't have host versus graft and other things like that. We'll talk about that. Organic disease, cancer, liver malfunction, diabetes. All right, so you have other concurrent comorbidities going on. So your immune system is already compromised, or your immune system's fighting other things. It can't take care of all of it. Very similar to, the environment right now, political. If we keep everyone fighting and doing all this stuff, it's much easier to gain hold if we've got everyone busy doing different things. Chemotherapy and immunosuppressant drugs. Let's just say somebody had bone marrow transplant or organ transplant, or their chemotherapy for cancer or other diseases. Physical or mental stress can really take a toll on your immune function, and any other infections you have are going to diminish your immune response. All right, so true pathogens are capable of causing disease in healthy persons with normal immune defenses. So these are pathogenic to most everybody. You can be super healthy, and these would be true pathogens. So it's not like it's opportunistic. Right, so influenza virus, plague bacillus, so bubonic plague or things, Yersinia pestis, things like that. Malarial protozoan, you get infected with one of these hemophalangites that's injected into your blood. It goes to your liver. It starts merozoites and starts producing all kinds of things. No matter how healthy you are, it's going to be a problem. Right. Opportunistic pathogens cause disease when the host defenses are compromised or when they grow in parts of the body that is not natural to them. All right, Pseudomonas, Candida albicans, this causes oral thrush, yeast infections. Pseudomonas, we will be talking about that. It's nasty. It grows on sponges. It grows on plumbing. It doesn't even need a carbohydrate source for energy. Anything with carbon, it can live. Right. Severity of the disease depends on the virulence of the pathogen. Characteristic and structure that contributes to the ability of the microbe to cause disease is a virulence factor. Right, so anything, we talked about slime layer, flagella, pili, and I guess you guys yesterday, what's the advantage of doing a flagella strain? Why do we want to know? Why do we want to know if something is a spore former? Why do we want to know if it has a slime layer? Why do we want to know if it is a capsule? Why do we want to know if Staph aureus can secrete enzymes that will dissolve hyaluronidase or coagulase? Can it clot? We'll see that today. It's going to clot the rabbit plasma, hopefully, if it works. Right. So we just did this in safety first lab. So we're literally here, we're just dealing with biohazard 1 or possibly biohazard 2. Right. I think the worst thing we have here might be E. coli, but we don't have like H1 and oh good Lord, 157H1. It's not hemorrhagic E. coli. It's just basic E. coli. So I think it's still bio level 1. We have Mycococci luteus, gram-positive. We're going to be dealing with the lactobacillus and Saccharomyces on Thursday when we start dealing with the molds and yeast, mycoses. Right. So becoming established. So ports of entry. Characteristic route of microbes follows enter tissues of the body. So exogenous agents originating from the source outside the body. So that's something you breathe in, you take in. Now, endogenous, you could have, if you're immune compromised, you could have something from inside your body that gets to the wrong area somehow. So you're self-inoculating. So whether it is you're self-inoculating after you've gone to the bathroom and you didn't wash your hands real well, you touched your eye or your nose or whatever, and you got something from your GI tract somewhere where it doesn't belong. All right, that's an endogenous agent. You self-inoculated. Right. So porous entry can be skin, nicks, abrasions, punctures, and incisions. And I asked, you guys asked me yesterday about staph and how it causes infections. Right. Usually, if it breaches your skin, that's a problem. All right. The other thing is, they found out that a lot of these organisms won't even grow that well in a cut, but the minute you put stitches on it, it has something to attach to, and now you've got a biofilm issue going on. Gastrointestinal tract, so food, drink, or other ingested materials. And so let's just say you have you eat something that has bacteria growing in it. You get sick from that. Or if you have Staph aureus, it may not even be multiplying. It might just be secreting exotoxins. Now you have a food intoxication, not food infection. Right? Respiratory tract, we have oral and nasal cavities. Even though you have IgA in there, you know, that mucous membrane is inflamed from a cold or a flu or something, it's much easier to breach that. Urogenital tract, sexual or displaced organisms. You know, we're not going to get into sexual diseases right away, but please realize that some urinary tract infections can be just from improper wiping, or fecal matter gets there, or, you know, if you have fecal matter in your reproductive tracts, so however you got there, can cause problems. Let's just leave it at that. Transplant placental. All right, so this is vertical transmission. This is where mom can infect the infant. There's a few diseases like listeria and some of the other ones that are transplacental. It also means vertical transmission. It's the same thing. When mom is harboring something and she inadvertently passes it on to the the baby. Storage, if you ever see that, these are just kind of pathogens that affect during pregnancy. So syphilis, toxoplasmosis, we'll talk about that with cats. Hepatitis, AIDS, chlamydia, rubella, cytomegalovirus, herpes simplex. Right. So infective dose. This just means that some things you only need is 1 or 2. It's an effective dose. Other things you need hundreds or thousands. So it's the minimum number of microbes required for infection to proceed. Microbes with small infective doses have greater virulence. Right? Lack of infective dose will not result in infection. So let's just say we'll use cholera, all right, for example, because this people, they pass this around in third world countries through the water or whatever. So let's just say you ingested 10,000 cholera cells. You won't get sick. You need that infective dose. All right. Tomaria, you can get that from rabbits or rodents out in the wild. Q fever, 1 to 4 cells. Tuberculosis is not up here. It's weird. Okay, so plague, gonorrhea. You need this certain, oh, bubonic plague right there. So you need a certain amount. If you don't have that threshold of that amount, you most likely will not get sick. I'm not going to say you won't. But if you look, measles just needs 1, Q fever. These are very virulent. It doesn't take much. Right. So we'll get into some other really cool stuff, or what I think is cool. So attaching to the host, adhesion, microbes gain a favorable foothold at the portal of entry, dependent on binding between specific molecules on the host and the pathogen. All right, so if we have fimbriae, these are like burrs, right? They can attach, and E. coli has this, and they can attach to the urinary tract. And like I said, they can crawl up the ureter. You can pee all you want. You're not gonna rinse this thing away. Vigella, we talked about how it can do the run or tumble. Glycocollax, so that's going to be your sugar coating. It can be almost like a slime layer. It causes things to stick, so they're harder to move. All right, doesn't help them with mobility. Cilia, Benelum coli through pig feces is the only one that is ciliated that would find a human host. It's mostly zoonotic. Suckers, hooks, and barbs. Those are going to talk about those when we talk about the tapeworms or worms. All right. So adhesion properties of microbes. Here, E. coli, well-developed fimbriae adhesions. They attach really, really well. Neisseria gonorrhea, double D. Fimbriae attached to the genital epithelium. Shigella, these are going to attach to the intestinal wall. Vibrio, all right. These are almost comma-shaped. And Trypodium, that is a spiral. It burrows right into your mucous membranes. Mycoplasm. All right, these literally they don't really have a certain shape, so they really kind of take on the shape of whatever. And they, a lot of surface area with those, they stick pretty well. Strep mutans, dental dextrin slime layer causes a biofilm. Right. Viruses usually have spikes. You know, everyone thinks that corona was the first one with spikes. It's not. Initial response to the host defense becomes form of phagocytes. So phago means eat. Cytes are usually grown cells. So these are large cells that eat. And like I said, they're going to look for PMP, so pathogen associated molecular patterns. They're not super specific. Anything that they find foreign, they're going to engulf it, right? They're going to break it down, try to digest it. Some of these are going to be antigen presenting cells. They're going to be presenting the antigen to the immune system. We'll talk about that later. Antiphagocytic fibers used to avoid phagocytosis. So we talked about capsules, and I talked about TB and how it has that waxy mycotic layer. So even if you get engulfed by a phagocyte, it's very hard for that phagocyte to engulf you and digest you. Species of Staphylococcus and Streptococcus produce leukocidins, and I mentioned these in lab. Leuko means white, toxic to white blood cells, and white blood cells we know are our immune system cells. So whether it's a neutrophil, basophil, lymphocyte or whatever, it can be very toxic to those. Slime layer or capsule makes phagocytosis difficult. And the ability to survive intracellular phagocytosis. So there are some bacteria we're going to learn about, like Listeria monogenesis. All right, they literally trick the monocytes into engulfing them. The monocyte can't kill them. Now the monocyte will Uber them anywhere they want in the body. All right, so pretty cool. Entering host tissue. So Shigella, Salmonella, these can trick the cells into engulfing them, and then they can either go to the adjacent cell, or they can go all the way to the basement membrane and get into the bloodstream. So, talked yesterday about virulence factors in lab. So virulence factors, traits used to invade and establish themselves in the host, also determine the degree of tissue damage that occurs, severity of the disease. And I said that Staph aureus has a lot of virulence factors, can secrete tremendous amount of enzymes that can deal with anything you throw at it, pretty much. Exo means out, exocytosis or whatever. Dissolve extracellular barriers and penetrate through or between cells. So this is the hyaluronidase. Some of these enzymes, they literally can, like I said, it will digest its way through anything in your body. Any of these membranes that separate things, it'll just eat its way through it. Toxogenicity, capacity to produce toxins at the site of multiplication, and they'll just secrete these toxins and they'll kill everything around it. All right. So there's two types of bacterial toxins, endo and exo. Remember those two words from A and P. Toxins that is not secreted but is released after the cell was damaged. This is generally gram negative, and they release lipopolysaccharide, and we talked in lab yesterday. I wanted to know the difference between gram positive and negative, and you guys were good and told me the gram negative is an outer membrane with the porins, a small peptidoglycan layer, and the other membrane. So when these gram negatives die, they release lipopolysaccharides, very, very toxic, causes intravascular coagulation, fever. It's part of the outer membrane of the gram negative cells, very, very toxic. Right? But it's only generally released after it dies, so we don't want to kill a lot of gram negative bacteria all at once. Exotoxin, toxin molecules secreted by a living bacteria cell into the infected tissue. So this is strong specificity for a target cell. So hemolysins, once again, these destroy red blood cells. I can grab heme or iron if I want to reproduce. We have AB toxins, the active site and the binding site. So it has to bind, and then it has to have active, so it can release the toxin in the tissue, causing the damage. And this is exo. So this is being released while the bacteria is alive and it's going to be a virulence factor. It's going to release to help it survive or make it more virulent. So exotoxins, target organs are damaged, heart, muscles, blood cells, intestinal tract show dysfunctions. You could have wicked diarrhea, start having secretory diarrhea. Endotoxins, like I said, these are the outer membrane. These release the LPS, lipopolysaccharide, causes general physiological effects, but always fever, malaise is that tired feeling, you don't know exactly what's going on, aches, shock, and that's the intravascular coagulation. All right, so your blood is clotting inside your blood vessels. Never a good idea. All right, so here's just some things to look through. All right, when you're going to start studying, or you want to make a card of this, or you want to print this out. So exotoxins are a tiny amount. All right, endotoxins are toxins at a higher dose, but this is after the microbe dies. That's gram-negative. All right, so effects in the body: gram-negative fever, inflammation, weakness, and shock is that intravascular coagulation. This is going to be more targeted, like blood, liver, nerve. All right. Chemical composition, small proteins. These are going to be the enzymes that they're going to be secreting. And the endotoxin, the gram-negative, is going to be that LPS, lipopolysaccharide. All right, here's just showing you the A active component is attached to the B binding. So it has to bind just to be able to get into the cell or trick that cell into opening. So we have to have, it's got to be very specific for the MHC or the marker on the cell. Right? The A component inhibits a cellular protein to cause a damage. That should play for you guys. I think it's embedded right. All right. Process of infection and disease. All right. So this is kind of, we talked about inhibitory, the lag stage, stationary phase, and the death phase. It's kind of similar to that. So the incubation period is time from initial contact with the infectious agent to the appearance of first symptoms. So we can inoculate, you become exposed to something. It's going to kind of sit there. It's got to make sure the environment's right, pH is right, temperature's right, food source is right. It sits there and it says, oh, you know what? This is probably a good place. Then it starts to multiply. Agents multiplying, but damage is insufficient to cause symptoms. Several hours to several years. Some diseases take years. All right. So we have the incubation period, and then it starts multiplying to the point where it's almost the height of infection. But this is when you wake up and you have that malaise and you're not quite sure what's going on. You don't have the full infection yet, but you know something's going on. Right? And I think we talked about this lecture 1, some of these terms. So prodromal, vague feelings of discomfort, non-specific complaints. Don't know exactly what's going on, but you know something's up. Period of invasion. This is when it's multiplying at high levels, becomes well established, more specific kinds of signs and symptoms. And the convalescent. Remember that term I said they used to call nursing homes convalescent homes? As a person begins to respond to the infection, symptoms decline. So this is the recovery period. So it takes a while to brew. It says this is a good thing. We want to start multiplying. Hasn't gotten to the critical point yet. So it's still prodromal. Then you get that height of your infection. Always generally levels off. All right, because the microbes are multiplying. They multiply so fast that now they start only multiplying as they die off. So we have that consistent number. And then at some point in time, they should all die off. Right? So localized infection, microbes out of the body and remains confined to a specific tissue. And that's kind of what you want. You don't want it spreading. You don't want it systemic. Right? So if it's a local infection, hopefully it's cutaneous or subcutaneous. It's pretty much sequestered. Systemic infection spreads to several sites in tissue fluids, usually in the bloodstream. That's never a good idea because it can get off anywhere it wants. Focal infection, when an infectious agent breaks loose from a local infection, is carried to other tissues. So you can have an infection All right, but it gets the infection releases so much damage or so many enzymes that it breaks through the basement membrane and breaks through whatever boundaries or barriers there are, and then it just spreads. So mixed infection. Remember, I told you guys, your patient can have ticks and fleas. There's never going to be usually one causative agent. And I said, well, why do we want a street plate? What I was trying to say, usually if you have an infection, it's going to be more than one thing. You want to streak it out so you can say, oh, well, I'm going to streak it out and I've got 3 different isolates here. I'm going to grow that colony, see what it is. I'm going to do a gram stain on all 3 of those and figure out I have a mixed polymicrobial infection. What's happening here? Primary infection is your initial infection, and usually you could have an initial infection. It causes damage to the area, and then other things can come along and they can set up shop super easy. Like I said, if you have the flu or things like that, or you have like pneumonia, generally you always had something going on that was compromising your upper respiratory tract or causing inflammation or keeping your immune system busy, and then something else came along. And I told you guys in lab yesterday that I've never had the flu. I don't know why. Okay, let's go on. Sensors. My allergies, I think I would, but all right. Patterns of infection. Remember, acute and chronic. Acute comes on rapidly with severe but short-lived effects. Chronic generally means it's long, it's chronic, it's for months, years, decades. But generally the signs and symptoms are a little less severe. I'll use that word for lack of something else. So common signs and symptoms of infectious diseases. All right. So these are signs, these are symptoms. All right. So signs are like if you're talking about a soap note, all right. Signs, everyone objective, everyone can see it. Symptoms are what's the patient telling you? It's a symptom. A sign is something that's reproducible, or everyone in the room should be able to pick up on that sign. So please realize that distinction. All right. Signs and symptoms of inflammation. Early symptoms of disease are a result of activation of body defenses. So the first line of defense is always inflammation. I don't care whether it is a burn, frostbite, cut, crushing wound, gunshot wound, or whatever. Always inflammation immediately. It's your body's first line of defense. Signs of inflammation are edema, accumulation of fluid. You want to extracellular fluid. You're going to leave the capillary beds. Capillary beds become permeable. That plasma leaves. We're going to be flushing out any toxins, hopefully. There's a reason behind all that. We're beginning protein to the area. Oh. Okay, guys, I got to pause this for a second.